This is the fifth part of our Fastidious gram-negative bacilli lecture, and in this lecture we are going to focus on the organism Brucella. The Brucella organisms are not related to any other genus. There are 6 species in the genus Brucella, and 4 of these cause human disease. And the Brucella species are differentiated based on what animal they normally reside in. So Brucella abortus is normally found in cattle. Brucella melitensis is found in sheep and goats. Brucella suis is found in swine. Brucella canis in dogs. Brucella ovis in sheep, and Brucella neotome in the desert wood rat. So Brucella causes the disease Brucellosis, and the 4 species that will cause brucellosis in humans are melitensis, abortus, canis, and suis. Now Brucella can produce a very small capsule, and virulent strains are able to grow and proliferate in the macrophages, in the phagocytic white blood cells in the body. So the diseases, again, we said brucellosis, also undulant fever and Malta fever. Those are different names for brucellosis. The symptoms, usually 7 to 21 days after infection, so that's a long incubation period. A person will feel very tired, get chills, have a fever. They'll get drenching sweats, body aches, headaches, won't be able to eat. And this is due to these recurring bacteremic episodes. So what happens with brucellosis is the organism gets into the bloodstream, causes a fever, chills, sweats, and then it starts to go away. And so then, the patient might be seeming a little bit better. And then all of a sudden it gets back into the bloodstream, and again, fever, sweats, chills. It can lead to splenomegaly or enlarged spleen, lymphadenopathy, swollen lymph nodes, hepatomegaly, enlarged liver. Osteomyelitis is also a common complication, so the organism will get into the bones. Now, in its natural habit, Brucella will infect animals, sheep, cattle, pigs, dogs. It will produce very mild symptoms in these animals, or it's completely asymptomatic. The Brucella tend to have a predilection for organs that are rich in erythritol. So in animals, these are the breast, the uterus, the placenta, and the epididymis. So in animals, brucellosis leads to sterility, abortions, and asymptomatic carriage. So an animal can just asymptomatically carry Brucella, just like some of us asymptomatically carry Staphylococcus aureus in our nasal passages. So same thing in animals. Transmission, you can have direct or indirect transmission. Direct transmission is usually an occupational transmission. So farmers, slaughterhouse workers, veterinarians who are in close contact with animals are going to be at higher risk. Indirectly, you can get infected by consuming contaminated animal products. Raw milk or unpasteurized milk or unpasteurized cheeses made from milk from an infected animal, goat cheese, cow milk cheese. So unpasteurized though. Also, Brucella is one of the organisms that has been associated with infecting laboratory workers. So the other organism we talked about, if you remember, was Neisseria meningitidis. So Brucella has caused serious infection in lab workers due to contact with the organism and inhalation. So risks of infection, coming into contact with animals, a laboratory accident, things like that. So to diagnose Brucella, it is a very small rod. It's categorized as a coccobacillus. It looks similar to Haemophilus. It does require long counter staining to even take in the safranin. So it tends to be very, very light pink in color. It is difficult to detect the organism in clinical specimens because it's very tiny. So in order to see it, you may need a fluorescent stain, a direct fluorescent antibody stain. These are non-motile and they do not form spores. The most common specimen types that you would get in the clinical lab that would contain Brucella would be a blood culture, a bone marrow biopsy, a lymph node aspirate, a cerebrospinal fluid, or an aspirate from an abscess. And here is a gram stain of Brucella, very, very light staining, tiny, teeny tiny rod. So in order to definitively diagnose Brucella, you really need to grow it up, culture it, isolate it. And blood is going to be, without a doubt, the most common specimen you would isolate Brucella in. The media, it does require more of an enriched media. It will grow on blood agar plates. It requires thymine, niacin, and biotin for growth. And it used to be inoculated in what was called a castanata bottle, which is a special container with media that contains all of these nice goodies that Brucella likes to grow in. Brucella abortus requires 10% CO2 for its initial isolation. So definitely needs CO2 to grow, which is why it's fastidious. It is slowly growing. Usually it takes about 2 to 5 days to see colonies on an agar plate. It may take up to 4 weeks. So a negative culture does not actually rule out a Brucella infection because it's difficult to grow and it takes a long time to grow. Usually in the clinical lab, you'll look at your plates after 24 hours. You might look at them again after 48 hours. In some cases, for some organisms, you will look at them longer. But in general, you may not keep cultures long enough in order to definitively rule out Brucella. Under the microscope, again, very tiny gram-negative coccobacilli on blood agar plates. It's a non-hemolytic, translucent, opalescent. It has a very opaly appearance to it if you've ever seen an opal stone. It is a non-fermentative organism. It's catalase and oxidase positive. It's differentiated due to its requirements for CO2. It is urease positive within 2 hours, so rapidly urease positive. It will produce hydrogen sulfide and it grows in the presence of dyes. So there used to be a test where you could put dyes, basic fusion, and thionin onto filter paper and lay that across an agar plate that had a lawn, inoculate a lawn of bacteria, and depending on which dye the organism grew around, you can differentiate the species of Brucella that way. So the dye, here's an example of the dye inhibition test where you have your filter paper that has basic fusion, you have your filter paper that has thionine, and on the top there's Brucella melitensis, in the middle it's Brucella abortus, and on the bottom it's Brucella suis. So you could see suis grows in thionine, and so does melitensis. Suis does not grow with basic fusion, but melitensis does. So you can differentiate using this dye inhibition test. However, this test is not done in a clinical laboratory, and in fact, in a clinical lab, Brucella wouldn't necessarily be speciated. If someone had a Brucella infection, it would be considered a very serious infection, and you wouldn't necessarily have to go down to the species level. There's serological tests. You can agglutinate to detect surface LPS antigens, and that way you can speciate it into Brucella abortus or Brucella melitensis. And you would want to definitely do confirmation tests in order to really be sure you have a Brucella. There are Brucellogen skin tests, sort of like a TB skin test, where you get a cell-mediated immune response, and you would take the protein extract, well, that's what's in the skin test reagent, a protein extract from the whole organism. So you're not actually putting the live bacteria under your skin. It's an extract of protein. And if you get a reaction, an inflammatory reaction at this where you do your skin test, it would indicate that you had a prior exposure to Brucella, but it does not necessarily mean right now you actually have brucellosis. Keep in mind that Brucella is considered a bioterror agent, so it is one of the organisms we will discuss throughout the semester that is on the agent for bioterror, and so that's why if you were to isolate a Brucella from a clinical specimen, it would be immediately reported to the Department of Public Health, and I'm sure that specimen would go to the State Health Department. It would go to the CDC for further workup, just to be sure, is this an infection due to someone being in close interaction with an infected animal, or is this an bioterror occurrence? So that's just one thing to keep in mind.